Swayam Prabha. Digital India. Educated India.
Welcome back. So we'll start as we discussed in the last lecture about the three major focus of bioprinting, that is are the like the or challenges. The next step for 3D bioprinting is the bioprinting technology, biomaterials and sources. This lecture will discuss three more points that those are also the focus for future research, and also by when we do that, then we can also take this 3D bioprinting in the next level. Like vascularization technique is one of the another very important aspect of bioprinting, and we have devoted like two full lectures on vascularized tissue printing, what is the need for vascularization, how vascularized tissue printing can be done, this thing. So we have understood now the importance of vascularization in the tissue bed, because vascular, that is the driving factor for all tissue and organs, by which actually we can actually develop this tissue. We can the tissue can grow, tissue organ can grow, and then the function can be developed. So vascularization, a well developed vascular tree is required for large tissues, because large tissues, they are a typical vascular tree is present for all different large tissues, if you as we have discussed before. Like there are, and there are also the vascular channels, or the blood vessels. They are of different dimensions, starting from like centimeter to millimeter to micrometer. Different sizes, different ranges are possible, and also we have also earlier discussed that what are the different, say, layers, like what are the different tissue structures were there. There were they were like a vascularity, like different types of layers, like as we mentioned earlier, tunica intima, tunica media, tunica adventitia. All these different layers are there for larger blood vessels, and for the capillaries there is a tunica intima, and then there is a this the other one cell types that is present, that is that's a pericytes that is stabilizing the cell, stabilizing the blood vessels. So all these things are very important for vascularization. So a well-developed vascular tree has to be developed, has required to be generated for large tissues. Otherwise, this thing cannot be controlled. As we are discussing that that well-developed vascular tree required for large tissues, and there is a vascular tree is very much important for generating a particular tissue or organ structure. Like there are different types of vascular tree present in different organs, and we need to actually mimic that thing during bioprinting for generating a vascularized tissue construct. And we have also earlier discussed that different levels or different scales of vascular tissue, like starting from aorta, arterial, capillaries, veins. All these different things are possible with different types of bioprinting modalities. But again, if we can truly recreate or reproduce that vascular tissue structure, giving like this, follow that particular branching pattern, and also a particular dimension of the tissue structure is that vascular structure is very important. And those are the points to be discussed, and those are the points to be uh, addressed in the future also, because vascularized tissue construct, though there are some examples are there where a vascular tissue can be developed, well, like the vascular bed or the vascular tree can be developed, or the vascular channels can be developed. But integration of the vascular channel to the bioprinted tissue or organ is where that is still now it is not fully realized, and their scope for further research is there, and if there are some groups already working on that thing. So, and also we have to may have to engineer the bioprinted constructs. That means that integration of that biovascular tissue within this bioprinted construct, and along with this, actually when we are printing that tissue or organ, along with that only, if we can print the vascular tree along with that printed tissue, then probably it can be well integrated within this tissue, within this bioprinted tissue constructs, and that can probably give better vascular means better outcome we can probably get from this vascularized tissue printing thing. So that is the another thing. Vascularized bioprinting of vascularized tissue construct is the focus of future research for researcher, and that can take up the bioprinting in the next level, because unless we can address this supply of nutrients and oxygen to the each and every cells present in the vascular bioprinted construct, that the bioprinted construct cannot be sustained, cannot mature, and so the cells will finally ultimately cells will die if we don't do that. So that's why we need to engineer the bioprinted construct in that way we can create the whole structure. And then capillaries and microvessels required for tissue perfusion, that is because that's why the cells can survive the tissue this thing. So we need to have this capillaries, microvessels, all these different things into the different tissue structures. Then they have capillaries, microvessels, all these different things are there. Those things have to be generated within the tissue bed, so that we can able to recreate that kind of that anatomical structures that is required for perfusion of media, perfusion of means in the tissue level, that is perfusion of blood and other things. So when we are culturing them, so that we can perfuse cell culture media through that microvessels or capillaries, so that all the cells present within the structure, they can get nutrition, they can get oxygen, and they can survive and sustain. Other than this, suitable mechanical properties for physiological pressures and for surgical connection, that is also very much required when you are trying to develop that vascular, that blood vessels. That blood vessels should have some properties, as we have discussed earlier, like this. They should have a proper appropriate burst pressure. So it should not be like, suppose if we connect that to the artery where there is a huge pressure during the systolic event, so then with that pressure there should not be a bursting of that blood vessels. They can actually work in that physiological pressure range very well. That means they can dilate and constrict, but they should not get damaged due to this in that pressure level. And also there should be proper, like this should be have we should be able to properly connect them to the pre-existing blood vessels, so that by surgical connection, that is very important. But most of the time the blood vessels, they fail at their surgical connections, because they are mostly the, if that is not perfect or proper, then the blood clot can form there itself. The thrombus can be formed there itself. So it can block basically by this thrombus, then the whole thing can get blocked. Other than this thing, that because that the surgical that connection, that is the weak point. From there also the blood can leak. So that can also, the blood can leak from there. So that can also lead to other complications. So that's why the one thing is this, that means the blood vessels, they should have a suitable mechanical properties, and they should able to use them at that physiological pressure level, and surgical connections of this thing as that is another very important thing. So vascularization is a major challenge in case of bioprinted constructs, but unless we solve this thing, unless we can come up with strategies to do that, or bioprinting project cannot get success. Means projects cannot survive, cannot be successful, and vascular tissue construct is the need for this thing. Next thing is innervation. As we know very well that most of our tissue, it is highly innervated, and by this way there is a feedback mechanism. By this way, the many things are getting controlled. The whenever there is a electrical stimulation, because of that thing, our most of our tissue and organ structure, they can function because of this thing. And unless we can create this nervous tissue, that is very much that tissue cannot function in a normal way. So that, like this, innervation is required for normal tissue function. The normal tissue function, that is innervation, is very required for that thing. And unless you do that innervation, the tissue cannot function. Maybe inducible after transplantation using pharmacological or growth factor signaling. So many times during this bioprinting process, when you develop a particular bioprinted tissue or construct, that time if we cannot recreate or reproduce that kind of nerve structure, then what we can do, you can use some other strategy, like maybe we can use some pharmacological agents or growth factor signaling is required to generate the nerve structures there. So that is very other than this thing, the nerve conduits, there are strategies by which bioprinting, by which nerve conduits can be fabricated, and that nerve conduits can help into to uh, transplant whenever, suppose there is a nerve cut or something, so whenever there we can put this kind of nerve conduits, and so that this electrical signal can continuously transmit through this thing to the go to the other areas. So that's why one strategy to tackle this issue, maybe you can use some agents where that can be done to, by which the nerve tissue, suppose means nervous tissue can be grown, can grow after the bioprinting. Simulation before transplants could be achieved using bioreactors. Like in bioreactors are mostly used for uh, to generate the physiological function in the particular tissue by simulating that the condition, by like the process by which they undergo in the physiological conditions, like for bone tissue, where the compression and relaxation, that is the most important thing for bone tissue to grow. For cartilage also, the compression and those things are very important. For cardiac tissue, electrical maturation, like the electrical signals, where the signals can be given there, by that time the heart muscle can contract. So that synchronized contraction of heart muscle is very important for this thing. So that kind of thing. So innervation, for innervation also, we can, that can be also simulated in some bioreactor before the transplantation. So by this way, the nerve tissue can grow, they can mature within the bioprinted construct. That can be addressed. So these are the things, like these are the things that should be done before actually the actual transplantation happen. So these things should be taken care of. Now maturation of the tissue construct is another important area, where if the, suppose as I mentioned before, after bioprinting, that is not actually, we cannot call them a tissue, that is just a mixed cluster of cells and materials together. But when they grow, they maintain a particular order, a hierarchy. When they grow, that time a tissue can be developed, where the function is of that particular tissue is established. That is required. So that needs maturation. The bioprinted construct needs maturation to achieve that kind of physiological function. So, but for that, time is required for that to assembly and maturation. That is very much important. And we need to understand that what is the process of this assembly and maturation of the particular tissue. And if we can reproduce, if we can do that, and we have to use probably some certain cases, some bioreactors, or some dynamic culture conditions is required for maturation of the tissue. That is very much important. That is very much required. Now bioreactors may be used to maintain the tissues in vitro. In vitro, because in static culture most of the time it has been seen that the tissue, they lose their functions, the physiological function, because the tissue in the in vitro condition, in static culture, they cannot mature. And if we, when we provide them the dynamic culture conditions, that is mimicking that or simulating that body conditions, physiological condition, then the tissue maturation is faster, and the tissue can be grown into a mature tissue, right? So that's why bioreactors are mostly used. The bioreactors are mostly used for different purposes, like either to provide a dynamic culture condition, to simulate the physiological, suppose the loading, unloading, stretching, and also other, suppose some signaling. All these things can be given in the bioreactors. So those things are done mostly by using some types of bioreactor, and thus bioreactors are also very much important for maintaining the tissues in vitro and for further maturation. Provide maturation factor as well as physiological stressors. Like certain cases, some factors can be given for further maturation, and certain cases like growth factors. Very many times certain tissue growth depends on application of uh, growth factor, a temporal delivery of growth factor at different stages. So that's why this matures. So these maturation factors are required, and we need to provide them those things. So that can be another steps, so that by which we can take this bioprinted tissue construct further for physiological, for further for maturation and for their use, particular use for use. And there are some physiological stressors, like as you were saying, probably some kind of mechanical signals, like probably suppose some stress or strain that can be given, cyclical loading, unloading can be given, stretching, bending, all these things, because the most of our many of our tissues undergo this kind of physiological, the cyclical loading, unloading, and also different other signals, like probably other things like maybe electrical signals. There are different, the blood flow, in the blood flows through the tissue, that can also give some kind of some kind of shear stress. So that should be blood, when the blood flows through that acts on the blood vessels, due to they generate some shear stress. And that it has been also seen that the shear stress also help in angiogenesis. So that kind of study, that kind of things can also be provided within the bioprinted tissue constructs, and that is very helpful for growth or maturation of that thing. Potential for implantation testing of constructs. Then the other thing is this pre-implantation testing is also very much important for this thing, whether that, by which we can understand whether the tissue is tissue growth happens or not, the tissue is matured for that to actually help in to actually reproduce that physiological function or not. So those things also very important, by which we can actually do this physiological potential for pre-implantation of the tissue testing of concept. Now let us understand. Now, till now we have discussed the strength of bioprinting, by which actually we can generate different types of tissue or organ structures. But if you see, till now no bioprinted constructs have been put into human trials yet. There is no clinical trials for any of the bioprinted tissue or organs because of various reasons. First thing is, there are technological reasons, because the technology is not that matured, and it cannot actually recreate all these different, that intricate microstructure, mesostructure, or even the nano or microstructure or microarchitectures of that particular tissue, right? So our technology has not matured when we can actually reproduce or recreate the functional human tissue or organs. So there is a technological limitations. There are also some regulatory challenges, where suppose if we print a particular tissue or organ, then we have to first address whether this tissue or bioprinted tissue organ is safe to use and effective to use or not. How can you check the safety of this bioprinted construct, because these are personalized products? How can you check the safety of this thing? There are no proper standards available in any of the countries. The regulatory bodies now, they are coming up with standards for how can we check the safety of this product, of this bioprinted tissue or organs, by which the evaluation methods can be developed, standards can be developed, so that we can test our products based on the standards, and then we can, if it is safe and efficacious, then it can be approved for further clinical use. So now that's why there are twofold. First, the technological limitation and also regulatory challenges. When both these things can be addressed, then only we can actually use these bioprinted products for clinical applications. But let us see the realistic picture. Anything like any two-dimensional tissues, like maybe cornea, skin, these are like very short term. We can actually try. Maybe we can in a short term, very in a year, within a few years, we can able to get this kind of structure, paper structure, that can be used for clinical applications. Hollow tubes can also be, probably they are also not very, uh, complex in term. They're not complex like the solid organs, but those hollow tubes can also be developed in a short to middle term, within a few years or some years. Hollow organs may take few times, maybe they may like it may in the midterm, like maybe in the 5 years or so, we can get the solid organs. Is typically they have very typical structure, as we have discussed earlier also. So it may take some more time for solid organs to be to develop and to use. So that is a long term. Now let us understand exactly how different tissues, how can they. I have taken this from this link. So please go through this link to understand this furthermore. Like probably few tissues like joint cartilage, vertebral disc, skin, all these things can be developed within 2 to 5 years time, and that can be used. Like peripheral nerves, meniscus tissues, all these things can be developed in a few years, and that probably that can be used for clinical application within 5 years or so. But so that these are very soon. This come under very soon category. In soon category, like few things like trachea, throat cartilage, heart valves, anterior cruciate ligament, Achilles tendon, all these things. These things can take up few years, like probably around 10 more years or so, before they can actually be used for clinical applications. But if you think about uh, complex tissues like solid organs like heart, liver, kidney, lung, other thing. So these things, it may take 10 to 20 years before this thing can actually be used for clinical applications. As I say, there are some technological limitations and also there are some regulatory challenges. But if you see, we need artificial tissues and organs. There is a denying in that, because there are huge number of patients waiting for this artificial tissues and organs, and we can actually save many patients if we can generate artificial tissues and organs. And but probably one day we'll definitely reach there where we can print artificial tissues and liver or any complex organs, and the doctors would be able to use them for clinical applications. So suppose if someone coming up with instance liver failure, where that liver has to be replaced, then the doctor can help tell the patient, don't worry, your liver is getting printed and it will be implanted on you soon. So thank you for attending this lecture. I will again meet on the in the next lecture. Thank you very much.